Figure 629. The shoot apical meristem of grape can be seen in 3 dimensions in this scanning electron micrograph. Notice the large leaf primordium. An older leaf primordium was removed. The phyllotaxy is established at the very apex. As the leaf primordium grows upward, it increases in thickness, establishing the bulk of the midrib. A row of cells on either edge of the primordium grows outward, initiating the lamina. As a result of their activity, the young leaf consists of a midrib and 2 small, thin wings. All cells in the wings are meristematic, and their division and expansion enlarge the lamina rapidly. In a compound leaf, 2 rows of loci initiate leaflets, which then grow like simple leaves. During lamina expansion, stomata, trichomes, and vascular bundles differentiate, and the petiole becomes distinct from the midrib. The entire pattern is completed while the leaf is still very small, much less than 1/10 its mature size. In many perennial plants, leaves are initiated in the summer or autumn before they mature. After they reach the developmental state just described, they become dormant, part of a resting terminal or axillary bud. During the next growth period, usually the following spring, the bud opens and the primordial leaves expand rapidly as each cell absorbs water into its vacuole and swells. Little or no mitosis or cell division may occur, only maturation, especially the synthesis of chlorophyll, cutin, and wax. As the leaves expand, the immature, exposed epidermis is very vulnerable, especially to insects. In many species of annual plants, the process is similar, with the embryo in the seed acting like the buds of perennial plants. They initiate leaves before the seed becomes dormant and dry, while it is still inside a developing fruit, and those leaves absorb water and expand rapidly during germination. Figure 630. The uppermost leaf primordium is the oldest, and its sides have begun growing out as the lamina of a simple leaf of grape. In this compound leaf, rather than forming a lamina, 6 leaflet primordia have been established. Monocots. Monocot leaves, like those of eudicots, are initiated by the expansion of some shoot apical meristem cells to form a leaf primordium. Apical meristem cells adjacent to the primordium grow upward along with it, becoming part of the primordium and giving it a hood-like shape. More apical meristem cells become involved until the primordium is a cylinder that completely or almost completely encircles the shoot apical meristem. This tubular portion grows upward as a sheathing leaf base, and the original conical leaf primordium, now located on one side of the top of the tube, gives rise to the lamina. The outer surface of the tube is a baxial epidermis, the inner surface is a daxial epidermis. Meanwhile, the shoot apex enlarges, forms new stem tissue, and initiates the next leaf primordium, which will develop as a tube inside the previous leaf's sheathing base. After the first leaf has grown to its full size, the next leaf continues elongating and emerges through the opening at the top of the previous leaf's tubular sheathing base. Later, another, younger leaf will emerge and so on. Figure 631. Low magnification transverse section through young, developing leaves of the grass Panicum effusum. The central round structure is a section of the shoot apical meristem, and the leaves encircle it. At higher magnification, you can see that all cells in the leaves are still small and cytoplasmic, and the central vacuoles are small. At this stage, all cells are still undergoing mitosis and cytokinesis. Veins are starting to differentiate. At an older stage, leaf veins are more distinct. The first protoxylem tracheary elements are mature in some veins, but most cells that will differentiate into tracheary elements are still enlarging. Figure 632. Buds of buckeye expanding in the spring. Bud scales are being pushed back and will upsize as new foliage leaves swell. The smaller bud on the right contains only leaves, and its apical meristem may be initiating new leaf primordia that will expand a little later. The larger bud on the left has both new leaves as well as flower primordia. In some monocots, the lamina becomes broad and expanded like a eudicot lamina, but grasses, lilies, and many others have linear, strap-shaped leaves that grow continuously, having no predetermined size. Their lamina grows by a meristem located at its base where it attaches to the top of the sheathing leaf base. The lamina's meristematic cells remain active mitotically, producing new cells that extend the leaf. Even if a grazing animal, a range fire, or a lawnmower destroys much of the leaf, the meristem forms more lamina. This type of regeneration is not possible in most leaves. The constant basal expansion in monocot leaves means that protoxylem and protophlome are constantly being stretched and disrupted in the basal meristem. New vessel elements and sieve tube members differentiate rapidly enough that conduction is never interrupted. Just above the basal meristem is a region where tissues differentiate. More primary xylem and phloem are initiated, as are stomata and other features. Higher above the basal meristem, tissues are mature. Differentiation is similar to that for stems but is oriented upside down in monocot leaves. Figure 633. Aphids and other sucking insects typically attack young leaves and stems that have not yet developed any protective sclerenchyma around the vascular bundles. Figure 634. In a monocot, after the center of the leaf primordium has been initiated, neighboring regions of the shoot apical meristem become active and contribute to the primordium. This results in a leaf base that encircles the stem, forming a sheath around it 150, 120. The leaf blade of this dendrobium orchid has developed from a leaf primordium tip that resembled the tip-in, and the sheathing base here developed from elongation growth of the small collar of leaf primordium tissue visible in. Alternatives. Box 6-3. Photosynthesis without leaves. Photosynthesis may occur in tissues other than foliage leaf mesophyll, for example, stem cortex and bark. Carrying out photosynthesis in these alternative sites has particular consequences. Bark. A small number of trees produce green, chlorophyllous photosynthetic bark. This is rare and occurs almost exclusively in desert-adapted trees that upsize their leaves during dry periods, after which they must rely entirely on their bark for sugar production. The trees, often called palo verde, produce cork cells in their bark like all trees do, but they also produce cells called phyloderm, which contain chloroplasts. Phyloderm is usually rather compact with few intercellular spaces, and very little of the cell walls are free surface, that is, surface that faces intercellular space and that can absorb carbon dioxide. Consequently, carbon dioxide neither diffuses rapidly through phyloderm nor is it absorbed quickly. Furthermore, the layer of phyloderm cells is covered by several layers of cork cells, which are impermeable to gases and thus block uptake of carbon dioxide. The green bark of these trees probably carries out, recycling, photosynthesis. All living cells of the tree trunk and branches respire and give off carbon dioxide just like we animals do, and the green bark can recapture it and convert it back to carbohydrate. In trees with non-photosynthetic bark, that carbon dioxide would simply leak out of the bark and be lost. Cortex. Young stems of almost all plants have a chlorophyllous cortex. Most stems are too narrow to provide much surface area and thus capture little light. Whereas stomata are virtually always present in foliage leaf epidermis, they may be lacking in stem epidermis. And if so, little carbon dioxide can be absorbed. Even if stomata are present, stem cortex, like phyloderm, is typically more compact than leaf mesophyll and thus does not permit rapid diffusion or uptake of carbon dioxide. Stem succulent plants. Many desert plants store water in enlarged, chlorophyllous stems. Stem succulents may produce large, thin, ordinary foliage leaves during the desert's brief moist period, abscising these leaves when drought begins, or they may produce only very tiny leaves. Either way, stem succulent plants usually depend on their chlorophyllous cortex for much or all their photosynthesis, and their cortex typically has many leaf-like features. Their stem epidermis has a high density of stomata, sometimes having as many stomata per square centimeter as leaf epidermis. The outermost layer or two of cortex cells may be very aerenchymatous, creating a tissue similar to spongy mesophyll, allowing carbon dioxide to diffuse rapidly away from stomata and deeply into the cortex. Chlorophyllous cortex often resembles palisade mesophyll, consisting of long columnar cells. Whereas palisade mesophyll is only rarely more than 1 or 2 cell layers thick, palisade cortex in stem succulents is often 5 to 10 cell layers thick, being as much as 1 to 1.5 millimeters thick. Any photosynthetic tissue must be well vascularized, but cortex typically does not have its own set of vascular bundles. This vascular bottleneck can be overcome by several alternative arrangements. If the cortex is narrow, its chlorenchyma cells are close to the stem's vascular bundles. If the stem has short internodes, then leaf traces are also close to each other and provide some vascularization to the cortex as they run out to the petiole base. The cortex of cacti is exceptional in that it does have its own particular vascular system called cortical bundles. This is a network of collateral vascular bundles that branch and anastomose, forming a three-dimensional reticulate venation. Cactus cortex is well vascularized, and no matter how rapidly it photosynthesizes, phloem cells are close by and can quickly load the sugars and transport them away. Figure B6-3 trunks and branches of trees called Palo Verde are green because their bark contains cells with chlorophyll. No stomata are present, so no new carbon dioxide can be absorbed from the air. Trunks and branches of saguaro cactus are green because the epidermis is still present and transparent, allowing us to see the green of the cortex cells inside. Stomata are present, and saguaros do bring in carbon dioxide from the air and convert it to carbohydrate. As a saguaro absorbs water after a rain, its parenchyma cells swell so the entire stem swells, the ribs flatten out and the epidermis is not torn. If it does not rain for many months, the cactus gradually loses water, despite its cuticle and wax, and shrinks. The ribs also shrink and become narrower. A ribbed body like this can change volume without changing surface area. Broad succulent stems store water, which causes them a problem not usually faced by foliage leaves: how to allow volume to increase while surface area remains constant. We animals generate new skin anywhere, anytime, which is a good thing. If we become overweight, our volume increases, as does our surface area, and our skin grows rather than tearing apart and revealing our insides. However, plants only generate new epidermis at shoot and root apices and in growing leaves and flowers. Stem succulents solve this problem by having an accordion-like, pleated surface. As water is stored and volume increases, ribs become wider. As water is lost, ribs become narrower. Volume changes dramatically but surface does not. In these alternative photosynthetic systems, water conservation is an important factor, so none of these alternatives is free to evolve to be as effective at photosynthesis as an ordinary foliage leaf is. Most modifications that increase water conservation decrease photosynthesis and vice versa. In moist habitats, water conservation is of little importance, so modifications that increase photosynthesis will increase survival of the plants even though they cause plants to lose water more rapidly. In contrast, modifications that conserve water in dry habitats are more valuable than those that allow maximum photosynthesis. Of course, desert plants are not free to maximize water conservation to the point where they do not photosynthesize at all. Morphology and anatomy of other leaf types. Succulent leaves. Numerous adaptations permit plants to survive in desert habitats, one of the most common being production of succulent leaves. This is characteristic of species in the families Crassulaceae, Portulaceae, and Isoaceae, among others. Succulent leaves are thick and fleshy, a shape that reduces the surface-to-volume ratio and favors water conservation. Some leaves are cylindrical or even spherical, the optimal surface-to-volume shape. The reduction in surface area, which is advantageous for water retention, has the automatic consequence of reducing the capacity for carbon dioxide uptake. Inside the leaf, the mesophyll contains very few air spaces, reducing the internal evaporative surface area and, in turn, water loss through stomata. A lack of air spaces also makes the mesophyll more transparent, just as pure water is more transparent than soap bubbles or foam, allowing light to penetrate farther into the leaf. Photosynthesis occurs more deeply than it would in the foliage leaves described earlier. In some members of the genera Lithops and Frithia, leaves are so translucent they act as optical fibers. The leaves are located almost completely underground, where it is cool and relatively damp. The exposed leaf tips allow sufficient light to enter and be conducted to the subterranean chlorenchyma. Although the plants live in a harsh desert in Madagascar and southern Africa, photosynthesis actually occurs in a rather mild microclimate. Figure 635 leaves of Senecio rotundifolia are spherical, giving them an optimal surface-to-volume ratio for conserving water. Leaves of the succulent Dinteranthus are hemispherical and attached to the opposite leaf, greatly reducing the exposed leaf surface. Interior tissues are mostly water storage parenchyma. Their low levels of chlorophyll result in low levels of photosynthesis, but a lack of water is a more significant danger for these plants. Each plant consists basically of 2 leaves and a microscopic stem. The root system is substantial. Dinteranthus and many related species have only 2 leaves at a time. As 2 new leaves expand, the existing 2 collapse and wither away. This agave has survived a wildfire in West Texas because its succulent leaves protected the stem from the heat of the fire. The center of the plant, and especially the shoot apical meristem, is still healthy. Containing so much water, the leaves heat up slowly, and before the innermost leaves became hot, wind had blown the fire farther on and the stem and apical meristem were saved. The leaves even kept the ground at the base of the plant cool, protecting the roots. Figure 636 Lithops, stone plant, is closely related to Dinteranthus. Both are in the family Isoaceae. Lithops also has only 2 leaves, but they are located almost entirely underground. The flat, translucent tips project above the soil and conduct light to the subterranean chlorenchyma. Sclerophyllous foliage leaves. Foliage leaves must produce more sugars by photosynthesis than are used in their own construction and metabolism, or the plant would lose energy every time it produced a leaf. This limits the amount of sclerenchyma in foliage leaves, and most leaves therefore tend to be soft, flexible, and edible. In some species, leaves have evolved which are perennial, existing on a plant for 2 or more years. With this extended lifetime and prolonged productivity, sclerenchymatous leaves are feasible, and their hardness makes them more resistant to animals, fungi, freezing temperatures, and ultraviolet light. Such plants are sclerophyllous and the leaves are sclerophylls. The sclerenchyma is often present as a layer just below the epidermis and in the bundle sheaths, although the epidermis itself can be composed of thick-walled cells. The cuticle is usually very thick, and waxes are abundant on leaves of many sclerophyllous species. Figure 637 leaves of barberry are tough and hard. It is difficult for insects to bite into them or lay eggs in them. However, the plant must invest considerable glucose to make the secondary walls of sclerenchyma cells, and the leaves must therefore photosynthesize longer before they reach their break-even point. Leaves of conifers. In almost all species of conifers, leaves are sclerophylls. They have a thick cuticle, and their epidermis and hypodermis cells have thick walls. Most conifer leaves contain unpalatable chemicals. Conifer leaves are always simple, never compound, and have only a few forms. Needles, either short or long, occur in all pines, firs, and spruces. Needles of longleaf pine can be 40 centimeters long, although most other species of pine have needles about 10 centimeters long. Small, flat, scale-like leaves form a shield-like covering on stems of junipers, cypresses, arborvitae, and others. In Agathis, Araucaria, and Podocarpus, all genera of the southern hemisphere, leaves are rather large, broad scales held away from the stem. Lengths up to 12.5 centimeters and widths of 3.5 centimeters have been measured in leaves of Podocarpus wallachianus. Conifer leaves are mostly perennial, remaining on the stem for many years. Consequently, the plants are evergreens. Needles of bristlecone pine live for at least 5 years. Their vascular bundles can produce new phloem each year, but no new xylem. The small-scale leaves of juniper also persist and are photosynthetically active for many years, and leaves of agathis and araucaria remain even on very old trunks. 3 conifers have annual leaves that are shed each autumn. Larches, bald cypress, and dawn redwood are all deciduous. Bud scales. One of the most common modifications of leaves is their evolutionary conversion into bud scales. In perennial plants, dormant shoot apical meristems are protected from low temperatures and the drying action of wind during winter by bud scales, which form a tight layer around the stem tip. Because their role is primarily protection, not photosynthesis, bud scale structure differs from that of foliage leaves. Bud scales are small and rarely compound, so mechanical wind damage is not a risk for bud scales. Their petiole is either short or absent because they must remain close to the stem and be folded over it. To be protective, they must be tougher and waxier than regular leaves. Bud scales frequently produce a thin layer of corky bark, at least on exposed portions, which provides greater protection than the simple epidermis of foliage leaves. Spines. Cacti have two types of leaves. The green cactus body has microscopic green leaves, and the clusters of spines are their axillary buds. Cactus spines are modified leaves of axillary buds. The succulent, moist cactus body would be an excellent source of water for animals were it not for the protective spines. As with bud scales, spines have a distinct structure related to their function. The soft, flexible blade of a photosynthetic leaf is useless as a protective device against herbivores, but spines have no blade and are needle-shaped. Mutations that inhibit lamina formation have been selectively advantageous. No mesophyll parenchyma or vascular tissue is present. The mesophyll instead consists of closely packed fibers. After fibers mature, they deposit lignin in their walls, which makes them hard and resistant to decay. The cells then die and dry out, hardening even further. Because cacti carry out photosynthesis in their stem cortex, loss of the leaf lamina was not selectively disadvantageous, but for plants that have no alternative photosynthetic tissues, mutations that cause loss of lamina are extremely disadvantageous. Spines of many other species have a similar structure. Figure 638 yucca leaves are excellent examples of sclerophylls. They are tough and fibrous, and the cuticle is thick. These features reduce the amount of light reaching the chlorenchyma, but plant growth is probably limited by scarcity of water rather than reduced photosynthesis. These leaves may function for 5 to 10 years or more. Bundles of fibers occur just interior to the epidermis, and the thick mesophyll surrounds many vascular bundles. Stomata occur in long, shallow grooves that run between fiber bundles. Even vascular bundles of these sclerophylls have fiber sheaths. Sclerophylls of Dracaena are very thick and have leaf veins throughout the mesophyll, unlike the single row in ordinary leaves. Leaf veins of Dracaena have extremely thick fiber sheaths. Figure 639 Many conifers, such as this lodgepole pine, have needle-shaped leaves. Incense cedar and many other conifers have scale-shaped leaves. Figure 640 Scale-shaped leaves in monkey puzzle tree are very large and persistent, living for many years, even as the branch enlarges and becomes woody. Figure 641 Needle leaves of pine are round in transverse section. Needles last for many years, producing more phloem each year. Pine needles are sclerophylls. They have thick-walled epidermal cells with a thick cuticle. They also contain resin canals. Figure 642 Bud scales of rhododendron fit together tightly during winter, protecting the enclosed apical meristem and young leaves and flowers. The bud scales of magnolia have a dense covering of hair. This improves the insulation. Tendrils. The tendrils of many plants are another form of modified leaf. Unlike photosynthetic leaves, tendrils grow indefinitely and contain cells that are capable of sensing contact with an object. When the tendril touches something, the side facing the object stops growing but the other side continues to elongate, causing the tendril to coil around the object and use it for support. A lamina would be detrimental, and none forms. Whereas many foliage leaves sense the direction of sunlight and reorient the lamina for maximum photosynthesis, tendrils respond by sensing solid objects and growing around them. Figure 643 The spines of this barrel cactus are modified leaves. The cluster of spines is actually an entire axillary bud. Cactus spines have a basal meristem. As new cells are formed, older cells are pushed upward. As they move out of the meristem, they fill their central vacuole with water, elongate, then deposit a thick secondary wall and differentiate into fiber cells. Figure 644 This tendril is a highly modified leaf. It has no leaf blade, and whereas foliage leaves stop growing after they reach a specific size, this tendril continues to grow. Leaves with crans anatomy. A distinct type of leaf anatomy occurs in plants that have a special metabolism called C. photosynthesis. These leaves lack palisade parenchyma and spongy mesophyll but have prominent bundle sheaths composed of large chlorophyllous cells. Surrounding each sheath is a ring of mesophyll cells that appear to radiate from the vascular bundle. These plants possess a mechanism of carbon dioxide transport that requires this special crans anatomy and adapts C plants to arid environments. Insect traps. The ability to trap and digest insects has evolved in several families. Insectivory has evolved in plants that grow in habitats poor in nitrates and ammonia. By digesting insects, plants obtain the nitrogen they need for their amino acids and nucleotides. Trap leaves can be classified as either active traps that move during capture or passive traps incapable of movement. The best known passive traps are the pitcher leaves of Nepenthes, Darlingtonia, and Sarracenia. Although the leaf appears highly modified, it is actually similar to many foliage leaves. It is thin, parenchymatous, and capable of photosynthesis. It has numerous stomata and vascular bundles as well as mesophyll containing parenchyma and chlorenchyma. The most significant differences are that the lamina is tubular rather than flat, and it secretes a watery digestive fluid. The epidermis in the digestive region must be absorptive rather than impermeable. Also, the throat of the pitcher contains numerous trichomes that point toward the liquid. It is easier for insects to walk in the direction of the trichomes, and they are thus led to their death. Leaves of sundew are active traps. They too have many features in common with foliage leaves, but their upper surface is covered with glandular trichomes that secrete a sticky digestive liquid. After an insect is caught on a single trichome, adjacent trichomes are stimulated to bend toward the victim, placing their digestive drops on it as well. The entire leaf blade curls around the insect so that many trichomes come into contact with it. The modifications in these leaves have been largely metabolic rather than structural. Trichomes and lamina must be able to sense and respond to the presence of an insect, carry on secretion and absorption, and unfold after digestion is complete. Figure 645 Nepenthes has elaborate pitcher leaves. They have an ordinary petiole and lamina, but the leaf tip is long, narrow, and pendant. Its extreme tip turns upright and develops into the hollow pitcher that contains a digestive mixture and an epidermis that can absorb nitrogen. The ultimate portion of the leaf tip is a broad flat roof that prevents rain from falling into the pitcher and diluting the digestive juices. Sundew leaves have many shapes. Those of Drosera capensis are long and narrow. Just after an insect is caught, the leaf curls and places numerous stalked glands on it. The drop at the end of each gland is both sticky and digestive. Once digestion is complete, the leaf uncurls and is ready for its next meal. Each half of the blade of a Venus flytrap leaf has 3 hairs. If an insect touches any 2, it stimulates the trap to close. Venus flytraps have leaves that are held flat, like most foliage leaves, but in Dionaea, this position is maintained only because motor cells along the upper side of the midrib are extremely turgid and swollen. When an insect walks across the trap, it brushes against trigger hairs. If 2 of these are stimulated, midrib motor cells lose water quickly, and the trap rapidly closes as the 2 halves of the lamina move upward. On the margins are long interdigitating teeth that trap the insect. Short glands begin to secrete digestive liquid. After digestion and absorption are complete, the midrib motor cells fill with water, swell, and force the trap open, ready for a new victim. At the next level. 1. Ecological modifications of foliage leaves. Thin, flat foliage leaves absorb sunlight and carbon dioxide, and they lose water. This shape gives a plant a large area of contact with the environment. The size and shape of leaves, their volume, and surface area all must be adaptive in the plant's habitat. It is common to see suites of characters in most plants of particular habitats. For example, we expect to find certain leaf characters in cold, alpine, windswept habitats, different ones in hot, rainy tropical habitats. Topics to search for are drip tips, succulent leaves, sclerophylls, spines, and insectivorous plants. 2. Evolution of leaves. Foliage leaves evolved at least 3 times, in mosses, in lycophytes, and in seed plants and ferns. We know a great deal about certain aspects of leaf evolution, much less about other aspects. Search on topics such as microphyll, lycophyll, megaphyll, euphyll, telome theory, and transfusion tissue. 3. Abscission. Some leaves remain on a plant after they have died, but leaves of other species have abscission zones and cut themselves away from the plant when they can no longer photosynthesize, either because they are too old or too stressed. Abscission zones are also present and important in flowers, fruits, inflorescences, and twigs. In bald cypress, leaves remain on twigs but whole twigs are abscised, taking the leaves with them. Abscission zones vary in their structure and placement, and also in the factors that control when they are active in causing an organ to fall. Summary. 1. Natural selection has resulted in the evolution of numerous types of leaves involved in photosynthesis, protection, support, water and nutrient storage, and nitrogen absorption. 2. Foliage leaves must be resistant to pathogens and stresses, must produce more sugar than was used in their construction, and must not act as sails. Modifications related to these functions may prevent foliage leaves from being optimally adapted for photosynthesis. 3. Foliage leaves typically consist of a blade with or without a petiole. The blade may be simple or either pinnately or palmately compound. 4. Leaf variability may involve distinct juvenile and adult leaves or distinct leaves on long shoots and short shoots. 5. An abscission zone typically contains a separation layer in which cell walls rupture and a protective layer whose cells form a corky leaf scar. 6. Internally, most foliage leaves have an upper palisade parenchyma rich in chlorophyll and a lower spongy mesophyll that allows gas circulation. Vascular bundles composed of primary xylem and phloem are usually arranged in a netted pattern in basal angiosperms and eudicots and a parallel pattern in monocots. Minor veins load sugars and unload water. 7. Leaves are initiated only at shoot apical meristems, beginning as small conical leaf primordia. Eudicot leaves generally have diffuse growth in all parts simultaneously. Most monocot leaf blades grow from a basal meristem. Important terms. Abscission zone, bud, scales, bundle, sheath, compound, leaf, lamina, leaf, blade, leaf, primordium, leaf, scar, leaf, traces, leaflets, mesophyll, midrib, minor veins, palisade, parenchyma, parallel venation, petiole, rachis. Reticulate venation, sessile leaf, simple leaf. Spongy mesophyll, stipules. Review questions. 1. Leaves carry out many roles in a plant's life. Give examples of leaves that do the following. A. Provide protection. B. Provide support. C. Provide storage. D. Obtain nitrogen. Are leaf structures and metabolisms that are selectively advantageous for one of these functions also advantageous for all of them? 2. What is the most obvious function of foliage leaves? What are some other functions that are often taken for granted? 3. Most leaves can be quite thin. Why? What would be the condition of lower layers in a thick leaf? 4. What is the stalk of a leaf called? What is the broad, flat, thin part? 5. Box 6-1, Plants and People, Leaves, Food, and Death, describes several ways that leaves affect animals. What is the name of the toxic chemical in leaves of poison hemlock and death camas? In leaves of tobacco? Can this poison be absorbed by the mucous membranes? Can it be passed across the placenta to an unborn baby? This chemical causes cancer of which parts of our bodies? 6. In many monocots such as grasses and irises, foliage leaves have a shape quite different from that of eudicot leaves. Their shape tends to be. They lack a petiole and instead have a that wraps around the stem. Name 3 monocots that have leaves that are unusual in that they appear to have petioles and laminas. 7. If a leaf has a blade that consists of one piece of tissue, we say the leaf is. If the blade consists of several pieces, however, we say it is a leaf. What type of leaf is shown for a rose in figure 6-7? For a Virginia creeper? 8. how can you distinguish between a compound leaf and a twig with several simple leaves? Assuming both a simple and a compound leaf have the same texture, which is more easily eaten by an insect larva? 9. Does a plant produce only one type of leaf during its entire life, or can some plants produce various types of leaf? Explain. 10. There are two types of venation in the leaves of flowering plants. Describe the type found in eudicot leaves. Describe the type in monocot leaves. 11. What does the abscission zone do in leaves? Would you guess that there are or are not abscission zones in flowers and fruits? 12. In what ways does the upper epidermis of a leaf differ from the lower epidermis? How are these structural differences adaptive? 13. This page is 22 centimeters wide and 27 centimeters tall. Calculate its surface area. If this page were a leaf with the same density of stomata as a leaf of sunflower, how many stomata would be on the upper side of the page and how many on the lower side? 14. The interior tissues of a leaf are called mesophyll. In most leaves, there is an upper layer of columnar cells called the and a lower portion of open, loose parenchyma. In Box 6-2, Botany and Beyond, Leaf Structure, Layer by Layer, figure shows the upper layer of columnar cells, but they do not look columnar. Why not? Figure in the box shows the lower parenchymatous layer. Can you tell what the shape of the intercellular spaces is from this two-dimensional micrograph? 15. Many leaves have a big, obvious vein that runs along their center, and many veins emerge from it. What is the large vein called, and can a leaf have more than one? The smaller veins that emerge from it? What are the very smallest veins of a leaf called? Which veins supply water directly to mesophyll cells, the biggest veins or the smallest? 16. What is a bundle sheath in a leaf? 17. Draw a cross-section of a foliage leaf and label each part. Be certain to show all the types of vascular tissues in the bundles. 18. Which parts of the leaf in question 17 would be emphasized and which would be reduced in each of the following? Tendrils, spines, bud scales, scale leaves of a bulb, and succulent leaves of a desert plant? 19. Most petioles are rather small, but they can be either massive or very long. List several examples of unusually large petiole. 20. Shoot apical meristems make small groups of cells that protrude upward and develop into leaves. What are these small protrusions of cells called? 21. Look at the cross-sections of leaf primordia in Figure 631a. Each one wraps completely around the shoot apical meristem. Can you explain why each leaf appears to surround the meristem? 22. How does the growth of eudicot and grass leaves differ? Which type would be more capable of recovering from attack by leaf-eating insects or grazing deer? 23. Dormant buds, such as the terminal and axillary buds of twigs in winter, consist of bud scales, young leaves, and leaf primordia. Which were formed by the apical meristem first and which were initiated last? 24. Figure 636 shows many plants of the desert plant lithops. How many leaves does each plant have? 25. Sclerophyllous foliage leaves like those in holly, barberry, and yucca. Have many unusual features. Do they live for 1 year or longer? They are more resistant to animals that might try to eat them. Why? What type of cells do they have that most leaves do not have? 26. Leaves of conifers are perennial. Those of bristlecone pine live for at least years, and their vascular bundles produce new every year. Many conifers have needle-shaped leaves, but those of incense cedar are shaped. 27. The spines of cacti are modified leaves, and as with bud scales, spines have a distinct structure related to their function. Why would the blade of a photosynthetic leaf be useless as a protective device? Cactus spines have no mesophyll parenchyma or vascular tissues. Instead, their mesophyll consists of. What chemicals do spine cells have in their walls? Are spine cells parenchyma, collenchyma, or sclerenchyma? After spine cells are mature, do they remain alive? Look at all your answers to this question. Does this modified leaf have much in common with a foliage leaf? 28. cacti have types of leaves. The green cactus body has green leaves, and the clusters of spines are there. Spines are modified leaves of axillary buds. 29. Several species have leaves that catch and digest insects, obtaining nitrogen fertilizer from their bodies. Describe how each of the following trap leaves function: the pitchers of Nepenthes, the flypaper leaves of sundew, the traps of Venus flytrap. Which are active and which are passive? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated ZArt. Chapter 7 Roots. Chapter Opener Image. Roots play many roles in the life of a plant, but a common function is to provide anchorage and stability. The roots shown here are old and woody, but they were initiated when the tree was just a sapling. It grew from a seed that had landed on the side of a fallen tree, so the young roots had to grow over the trunk of the dead tree for more than a meter before they ever reached the soil. In several decades, the fallen tree will decompose completely, then the roots will be holding up the trunk of their tree, more than a meter above the ground with just air between them. Outline. Concepts. External structure of roots. Organization of root systems. Structure of individual roots. Internal structure of roots. Root cap. Root, apical meristem. Zone of elongation. Zone, of maturation root hair zone. Mature portions of the root. Origin and development of lateral roots. Other types of roots and root modifications. Storage roots. Prop roots. Aerial, roots of orchids. Contractile roots. Mycorrhizae. Root, nodules and nitrogen fixation. Hostorial roots of parasitic flowering plants. Roots of strangler figs. Box 7 to 1 Plants do things differently, plants and people and having a weight problem. Learning objectives. After reading this chapter, students will be able to list the 3 main functions of roots. Explain the properties and biological consequences of taproots, lateral roots, and fibrous root systems. Describe the structure of individual roots. Name the components of the internal root structure. Compare lateral root development to bud formation in shoots. Discuss other types of roots: storage, prop, aerial, contractile, mycorrhizae, nodules, hostorial, and strangler fig roots. Did you know? We know very little about roots because they are hidden in soil and we cannot easily see when they are active or dormant, or even if they have died. Root organization is similar to shoot organization. Roots have an epidermis, cortex, phloem, and xylem, but they lack pith. Certain cells in the tips of roots have dense starch grains that sink to the bottom of the cells. This indicates to the cells which way is down. Some plants that grow along seashores have roots that grow upward, providing submerged roots with oxygen. Concepts. Most roots have 3 functions: anchoring the plant firmly to a substrate, absorbing water and minerals, and producing hormones. Firm anchoring provides stability and is therefore important for virtually all plants. Stems, leaves, flowers, and fruits then can be properly oriented to the sun, pollinators, or fruit distributors. Without proper root attachment, trees and shrubs could not remain upright, and epiphytes would be blown from their sites in the tree canopy. A highly branched rhizomatous or stoloniferous plant might resist being blown over even without roots, but their horizontal stems are usually so flexible that roots are necessary to stabilize the aerial structure. Although roots, like leaves, have an absorptive function, the 2 organs have totally different shapes. Sunlight always comes from above, but water and minerals are distributed on all sides of a root. Its cylindrical shape allows all sides to have the same absorptive capacity. Consider a leaf and a system of thin roots, both with equal volumes. The root system has a higher surface-to-volume ratio, ideal for absorption. The lower surface-to-volume ratio for leaves reduces the carbon dioxide absorption capacity, but not the absorption of light, and is actually beneficial for water conservation. Roots do not need to be adapted for light absorption, and they absorb water rather than needing to conserve it. Their cylindrical shape is also undoubtedly related to the growth of the roots through a semi-solid, resistant medium. Even a light, porous soil can be most easily and thoroughly penetrated by narrow cylinders rather than thin sheets. Thus, although both leaves and roots have absorptive functions, it is selectively advantageous for them to have different shapes. Roots are quite active in the production of several hormones. Shoot growth and development depend on the hormones cytokinin and gibberellin imported from the roots. This reliance of the shoot on root-produced hormones may be a means of integrating the growth of the two systems. It is selectively advantageous for a plant to control the size of its shoot so that transpiration by its leaves does not exceed absorption by its roots, and a plant should not waste carbohydrates by constructing a larger root system than its shoot needs. The extra carbohydrate could be used for leaves or reproduction. In many cases, roots have functions in addition to or instead of anchoring, absorption, and hormone production. Fleshy taproots, such as those of carrots, beets, and radishes, are the plant's main site of carbohydrate storage during winter. As roots of willows, sorrel, and other plants spread horizontally, they produce shoot buds that grow out and act as new plants. This method of vegetative reproduction is quite similar to that of stoloniferous and rhizomatous plants, except that roots rather than stems are involved. In the palms Chrysophila and Mauritia, roots grow out of the trunk and then harden into sharp spines. Ivy and many other vines have modified roots that act as holdfasts, clinging to rock or brick. Finally, many parasitic flowering plants attack other plants and draw water and nutrients out of them through modified roots. Distinct sets of characteristics are adaptive for different root functions. As roots specialize and become more efficient for particular tasks, they become poorly adapted for other tasks, and thus, several types of roots may occur in one plant, resulting in division of labor. For example, in addition to the large storage root, carrots and beets also have fine absorptive roots, and ivy has both holdfasts and absorptive roots. The characteristic types of structure and metabolism of each should be analyzed in terms of the function of the particular root. Figure 7.1 The ratio of root system to shoot system is critically important, for each species, as the root system becomes heavier, so does the shoot system by a characteristic amount. However, the relationship differs for each species. Obviously, for a given amount of turnip root there is relatively little shoot, but there is much more shoot for the same amount of pea root, and even more for cotton. External structure of roots. Organization of root systems. Roots must have an enormous absorptive surface. In order for a single unbranched root to have sufficient surface area, it would have to be hundreds of meters long, which would make conduction impossible. Instead, plants have a highly branched root system. Most seed plants have a single prominent taproot that is much larger than all the rest and numerous small lateral roots or branch roots coming out of it. This taproot develops from the embryonic root, called the radicle, that was present in the seed. After germination, it grows extensively and usually becomes the largest root in the system. Carrots, beets, turnips, and other taproots sold in stores have dozens of fine lateral roots while growing, but these are removed before the products are shipped to market. Table 7.1 Dimensions of the root system of barley, 4 weeks old. Figure 7.2 Some taproots, such as a carrot, become extremely swollen and are much larger than the numerous lateral roots, whereas in other species, such as sunflower, the taproot is about the same size as the laterals. The important criterion is that the taproot develops from the root of the embryo, the radicle. A fibrous root system, as in this winter wheat, consists of many roots, none of which is the radicle. Instead, all of the main roots are adventitious roots that originated in stem tissue. Figure 7.3 The taproot of this tobacco seedling is definitely larger than the lateral roots, but more importantly, its development can be traced directly to the embryo. Relative size is not the critical factor. In many species, lateral roots are the ones that become enlarged. The roots of radishes are obviously taproots. Lateral roots may also produce more lateral roots, resulting in a highly ramified set of roots analogous to the highly branched shoot system of most plants. Lateral roots can become prominently swollen like a taproot, as in sweet potatoes and the tropical vegetable manioc. If the plant is perennial and woody, roots also undergo secondary growth, producing wood and bark. Most monocots and some eudicots have a mass of many similarly sized roots constituting a fibrous root system. This arises because the radicle dies during or immediately after germination. Root primordia at the base of the radicle grow out and form the first stages of the fibrous root system. As the plant ages, more root primordia are initiated in the stem tissue. Because these roots do not arise on pre-existing roots and because they are not radicles, they are known as adventitious roots. Adventitious roots increase the absorptive and transport capacities of the root system. The functional significance of taproots versus fibrous root systems becomes apparent and the general growth forms of eudicots and monocots are considered. Many eudicots are perennial and undergo secondary growth, resulting in an increased quantity of healthy, functional wood in both the trunk and roots. This enlarging conduction capacity permits an increase in the number of leaves and fine, absorptive roots. This also occurs in other groups of seed plants such as conifers and cycads. Most monocots cannot undergo secondary growth. After their stem is formed, the number of vascular bundles, tracheary elements, and sieve tubes is set, and their conducting capacity cannot be increased. Extra leaves could not be supplied with water, nor could their sugar be transported. Such a shoot could not supply sugars to an ever-increasing taproot system. However, some monocots do increase their size by means of stolons or rhizomes. Their horizontal shoots branch and then produce adventitious roots. Because these roots are initiated in the new stem tissues, they transport water directly into the new portions of the shoot, unhindered by the limited capacity of the older portions of the shoot. By this mechanism, monocot shoots can branch and grow larger, as long as they remain close enough to the substrate to produce new adventitious roots. For them, a fibrous root system is functional, whereas a taproot system is not. The ability to form adventitious roots is not limited to monocots. Many rhizomatous and stoloniferous eudicots also grow this way naturally. Furthermore, many eudicots that never produce adventitious roots in nature do so if they are cut. This is important in the process of asexual propagation by cuttings. Figure 7.4 Corms of gladiolus, like other monocots, have a fibrous root system. The radicle died shortly after germination. No root here has developed from the radicle. As the gladiolus continues to grow, it will produce more roots, each independent of the others. Figure 7.5 A seedling of a woody plant has a few leaves and a small root system. The narrow trunk with a few vascular bundles can conduct water and nutrients between them. An older woody plant has more leaves and a larger root system. The stem has more wood and bark, which increases its capacity to conduct water and sugar. Because most monocots do not undergo secondary growth, the stem of an older monocot is not wider than that of a young plant, and it has no increased conducting capacity. Consequently, the old plant has no more leaves or roots than the young plant. If a plant can produce adventitious roots, the bottleneck of the monocot stem does not matter. New roots originate near the aerial shoots and conduct directly into them, and little or no long-distance conduction occurs in the rhizome. No part of the monocot stem needs to conduct all of the water from all the roots to all the leaves and flowers, as does the trunk of a woody plant. Figure 7.6 The tip of a root consists of a root cap, the root apical meristem, a zone of elongation growth, and a region where root hairs are formed. This radish seedling is only 3 days old and has abundant root hairs. Those near the root tip are short because they are newer and younger than those near the shoot. Structure of individual roots. An individual root is fairly simple, because it has no leaves or leaf scars. It has neither leaf axils nor axillary buds. The root tip, like that of the shoot, is the region where growth in length occurs. In roots, growth by discrete apical meristems is the only feasible type of longitudinal growth. In most animals, all parts of the body grow simultaneously, whereas roots and stems elongate only at small meristematic regions. Because the root is embedded in a solid matrix, it is impossible for all parts to extend at once. The entire root would have to slide through the soil. With apical growth, only the extreme tip pushes through soil. Figure 7.7. Although the exterior of a root appears rather uniform, several distinct zones of differentiation are present internally. Root hairs form only above the elongation zone, and the endodermis and first vascular tissues appear earlier than do root hairs. Whereas the shoot apical meristem is protected by either bud scales or young, unexpanded foliage leaves, the root apical meristem is protected by a thick layer of cells, the root cap. Although some soils appear soft and easily penetrable, on a microscopic scale, all contain sand grains, crystals, and other components that can damage the delicate apical meristem and root cap. Because the cap is forced through the soil ahead of the root body, it is constantly being worn away and must be renewed by cell multiplication. Figure 7.8. The root cap is distinct from the root proper. The root apical meristem is located at the apex of the root proper but is buried under the root cap. Because no leaves, leaf traces, or branches are present, cells develop in an extremely orderly fashion in regular files. It is easy to see that these cells are derived directly from the meristem, thus, they make up primary tissues. Screw pine has giant aerial roots that allow us to see the root cap more easily than with ordinary tiny roots. The root cap cells die and are sloughed off. Dictyosomes of root cap cells secrete a complex polysaccharide called mucilage, which lubricates passage of the root through the soil. It also causes soil to release its nutrient ions and permits the ions to diffuse more rapidly toward the root. Mucilage is rich in carbohydrates and amino acids, which foster rapid growth of soil bacteria around the root tip. The metabolism of these microbes is believed to help release nutrients from the soil matrix. Just behind the root cap and root apical meristem is a zone of elongation only a few millimeters long within which the cells undergo division and expansion. Behind it is the root hair zone, a region in which many of the epidermal cells extend out as narrow trichomes. Root hairs form only in a part of the root that is not elongating, otherwise, they would be shorn off. Root hairs greatly increase the root's surface area. In a study of rye, a single plant was found to have 13 million lateral roots with 500 kilometers of root length and a surface area of 200 m. Because of the abundant production of root hairs, however, the total surface area was doubled. Root hairs have other effects that should not be overlooked. Most pores in soil are too narrow for a root to penetrate. Root hairs, however, being only about 10 millimeters in diameter, can enter any crevice and extract water and minerals from it. Furthermore, carbon dioxide given off by the respiration of root hairs combines with soil water to form carbonic acid, which helps release ions from the soil matrix. Without the acid, ions would be too firmly bound to soil particles for the root to absorb them. Root hairs are unicellular, never have thick walls, and are extremely transitory. They die and degenerate within 4 or 5 days after forming. Figure 7.9. Much of a soil is composed of spaces between soil particles. In the uppermost 20 centimeters of a soil composed of sandy loam, 54% of the soil is pore space. Of this, 9% consists of extremely fine spaces less than 0.2 μm wide, 33% of spaces ranging between 0.2 and 60 micrometers, and 12% of large spaces wider than 60 micrometers. At deeper levels, the soil is more compacted, containing only about 42% pore space. The decrease is due mostly to a compaction of intermediate-sized spaces. Behind the root hair zone is a region where new lateral roots emerge. They may occur in rows or may appear to be randomly distributed on the parent root. Outgrowth of lateral roots often depends on the soil microenvironment. If the parent root grows into a zone of rich, moist soil, numerous lateral roots form, and the pocket is fully exploited. If the soil is poor, hard, or dry, few lateral roots emerge. Internal structure of roots. Root cap. To remain in place and provide effective protection for the root apical meristem, the root cap must have a specific structure and growth pattern. The cells in the layer closest to the root meristem are also meristematic, undergoing cell division with transverse walls and forming files of cells that are pushed forward. Simultaneously, cells on the edges of this group grow toward the side and proliferate. Although cells appear to extend around the sides of the root is actually growing through the edges of the root cap. Cells are small and meristematic when first formed at the base of the root cap, but as they are pushed forward, they develop dense starch grains and their endoplasmic reticulum becomes displaced to the forward end of the cell. These cells detect gravity because their dense starch grains settle to the lower side of the cell. As cells are pushed closer to the edge of the cap, their structure and metabolism change dramatically. Endoplasmic reticulum becomes less conspicuous, starch grains are digested, and the cell's dictyosomes secrete copious amounts of mucilage by exocytosis. Simultaneously, the middle lamella breaks down and releases cells, which are usually crushed by expansion of the root. It has been estimated that only 4 or 5 days pass from cell formation in the root cap to its sloughing off. Consequently, the cap is constantly regenerating itself. A dynamic equilibrium must be maintained between these two processes. Figure 7.10 in the root cap, cells in the central portion divide so that the two daughter cells are aligned with the root and the root cap grows forward. On the edges, cells divide and expand in such a way that cells flow radially outward. Root apical meristem. If the root apical meristem is examined in relationship to the root tissues it produces, regular files of cells can be seen to originate in the meristem and extend into the regions of mature root tissues. The root is more orderly than the shoot because it experiences no disruptions because of leaf primordia, leaf traces, or axillary buds. Because cell files extend almost to the center of the meristem, one might assume that cell divisions are occurring throughout it. However, the use of a radioactive precursor of DNA, such as tritiated thymidine, can demonstrate that the central cells are not synthesizing DNA, their nuclei do not take up the thymidine and thus do not become radioactive. This mitotically inactive central region is known as the quiescent center. These cells are more resistant to various types of harmful agents such as radiation and toxic chemicals, and it is now believed that they act primarily as a reserve of healthy cells. If part of the root apical meristem or root cap is damaged, quiescent center cells become active and form a new apical meristem. After the new meristem is established, its central cells become inactive, forming a new quiescent center. Such a replacement mechanism is extremely important because the root apex is probably damaged frequently by various agents, sharp objects, burrowing animals, nematodes, and pathogenic fungi. Figure 7.11. These roots were grown briefly in a solution of tritiated thymidine, a radioactive precursor of DNA. In cells that were undergoing the cell cycle, during S phase, the radioactive thymidine was incorporated into the nuclei. After a few hours, the root was killed, sliced into sections, and placed on photographic film in the dark. The radioactive nuclei caused black spots to form in the film next to the nuclei. The slide was then given a brief exposure to light so that the outlines of the cells and nuclei would be faintly visible. The quiescent center is the region where no nuclei became radioactive. Apparently no cell in the region passed through S phase while tritiated thymidine was available. These cells were in cell cycle arrest. Zone of elongation. Just behind the root apical meristem itself is the region where cells expand greatly. Some meristematic activity continues, but mostly cells are enlarging. This zone of elongation is similar to the shoot's subapical meristem region. Cells begin to differentiate into a visible pattern, although none of the cells is mature. The outermost cells are protoderm and differentiate into epidermis. In the center is provascular tissue, cells that develop into primary xylem and primary phloem. As in the stem, protoxylem and protophlome, which form earliest, are closest to the meristem. Farther from the root tip, older, larger cells develop into metaxylem and metaphlome. Between the provascular tissue and the protoderm is a ground tissue, a rather uniform parenchyma that differentiates into root cortex. In the zone of elongation, tissues are all quite permeable. Minerals penetrate deep into the root through the apoplast simply by diffusing along the thin, fully hydrated young walls and intercellular spaces. This zone is so short that little actual absorption occurs there, and much that is absorbed is probably used directly for the root's own growth. Zone of maturation. Root hair zone. In the maturation zone, several important processes occur more or less simultaneously. Root hairs grow outward, greatly increasing absorption of water and minerals. In some electron micrographs, a thin cuticle appears to be present on root epidermal cells, but this may be just a layer of fats. The zone of elongation merges gradually with the zone of maturation. No distinct boundary exists because the differences between the two represent the gradual continued differentiation of the cells. Although cortex cells continue to enlarge, their most significant activity is transfer of minerals from the epidermis to the vascular tissue. This can be either by diffusion through the walls and intercellular spaces or by absorption into the cytoplasm of a cortical cell and then transferal from cell to cell, probably through plasmodesmata. Cortical intercellular spaces are also important as an parenchyma, allowing oxygen to diffuse throughout the root from the soil or stem. Figure 7.12. The cortex may be broad in many roots, and its interior boundary is the endodermis. In the zone of maturation, minerals do not have free access to the vascular tissues because the innermost layer of cortical cells differentiates into a cylinder called the endodermis. Cells of the endodermis have tangential walls, those closest to the vascular tissue or the cortex, which are ordinary thin primary walls. However, their radial walls, the top, bottom, and side walls, that is, all the walls touching other endodermis cells, are encrusted with lignin and suberin, both of which cause the wall to be waterproof. The bands of altered walls, called Casparian strips, are involved in controlling the types of minerals that enter the xylem water stream. Cortex cells cannot exert any control over movement of minerals within intercellular spaces. Without an endodermis, minerals of any type could move from the soil into the spaces, then into the xylem, and then into all parts of the plant. Because Casparian strips are impermeable, however, minerals can cross the endodermis only if endodermal protoplasts absorb them from the intercellular spaces of the cortex apoplast or from cortical cells and then secrete them into the vascular tissues. Many harmful minerals can be excluded by the endodermis. It is not a perfect barrier against uncontrolled apoplastic movement because in the zone of elongation, where the endodermis is not yet mature, minerals do have free access to the protoxylem, but this is only a small amount of uncontrolled movement. Many glands and secretory cavities also have Casparian strips, which prevent the gland's secretory product from seeping into the surrounding tissues. Within the vascular tissues, many of the much larger cells of the metaxylem and metaphlome become fully differentiated and functional in the zone of maturation. The arrangement of these tissues differs from that in stems. Instead of forming collateral bundles containing xylem and phloem, the xylem of almost all plants except some monocots forms a solid mass in the center, surrounded by strands of phloem. No pith is present. In roots of many monocots, strands of xylem and phloem are distributed in ground tissue. Within the xylem, the inner wide cells are metaxylem and the outer narrow ones are protoxylem. 2 to 4 or more groups of protoxylem may be present, the number being greater in species with larger roots with a wider mass of xylem. The number of phloem strands equals the number of protoxylem strands. Within phloem strands, protophlome occurs on the outer side, and metaphlome on the inner side. Other than the arrangement, the vascular tissues of the root are similar to those of the stem and leaf. Those formed first are narrowest and most extensible, often finally being torn by continued elongation and expansion of cells around them. Those formed after adjacent cells have stopped expanding are larger and, in xylem, have heavier walls, often with bordered pits. Figure 7.13. The endodermis is a cylinder, one layer of cells thick, each with Casparian strips. The best analogy is a brick chimney. It is a cylinder, one layer thick. The cement is analogous to the Casparian strips. No apoplastic transport occurs across the endodermis. Materials that have been moving apoplastically are stopped and can proceed farther only if the endodermis plasma membrane accepts them. Symplastic transport is not affected. Plasma membranes that are impermeable to a particular type of molecule prevent that molecule from crossing the endodermis. The endodermis in this corn is relatively easy to see. In all cells, Casparian strips are visible as red lines on the radial walls. In a few cells, only Casparian strips are present, but in many, the radial walls and inner tangential walls have started to thicken. It is unusual for this development to start so early. Figure 7.14 Low magnification view and high magnification view of a transverse section of a Eudicot root. Showing the broad cortex and the small set of vascular tissues. The xylem has 3 sets of protoxylem, the narrow cells at the tips of the arms. The central, larger xylem cells are metaxylem. 3 masses of phloem are also present. This is a triarch root. The pericycle is the set of cells between the endodermis and the vascular cells, 50, 250. Low magnification view and high magnification view of a transverse section of a monocot root. The vascular tissue consists of numerous large vessels and separate bundles of phloem. The endodermis is very easy to see, 15, 150. Figure 7.15 The number of strands of protoxylem in a root varies from one species to another and among the roots of one plant. Generally wider, more robust roots have more protoxylem masses. Having 2 masses of protoxylem is diarch. 3 is triarch, and 4 is tetrarch. These diagrams represent 3 separate roots, not young and older stages of a single root. A diarch root does not become triarch and then tetrarch as it ages. Between the vascular tissue and the endodermis are parenchyma cells that constitute an irregular region called the pericycle. When lateral roots are produced, they are initiated in the pericycle. Plants do things differently. Box 7:1 Plants and people and having a weight problem. Most of us probably do not spend too much time worrying about starving to death. However, throughout much of the history of civilization, people had to be careful to store up enough food to last not only through winter but through spring as well. Food supplies had to last until gardens could provide potatoes, beans, wheat, and other staples. Grocery stores are a recent luxury. How did people store food? If dry seeds like wheat and beans are kept free of moisture, they last a long time. Grapes and milk are not dry, but they can be preserved by turning the first into wine and the second into cheese. Meat and fish can be dried, smoked, or salted for long-term storage. One particular method of storing food is particularly popular with humans and is the only means available to most animals. Eat the food whenever it is available, and store it as fat inside our body. Beans can become moldy. Rats can find our supply of dried meat, but fat in our adipose tissues is safe. For animals that hibernate, getting fat in autumn is the only way to survive. We may regret eating so much at Thanksgiving, but the Pilgrims did not. Feasting was a means of storing food that might otherwise spoil during winter. Plants too must be adapted to the availability, or the scarcity, of food. For plants, food is supplied by photosynthesis, and that requires only light, carbon dioxide, and water. In tropical climates where temperatures are always mild and droughts virtually never occur, plants have leaves throughout the year and photosynthesize every day, making all of the carbohydrates they need whenever they need them. Storing food reserves is not a problem. In temperate climates, evergreen trees such as pines and hollies also are able to photosynthesize most days, being inhibited only when it is extremely cold. But deciduous plants, those that drop their leaves and become dormant, are similar to animals in that they need to store food to maintain their metabolism while leafless. When a plant upsizes its leaves in autumn, it is almost as if an animal were throwing away its entire digestive tract in anticipation of growing an entire new one in spring. If a plant had no nutrient reserves inside itself when it upsized its leaves, it would not even be able to make new leaves in the following spring. It would starve to death. Energy reserves can be stored as a variety of chemical compounds. We animals sequester our reserve energy as fats. A little bit of energy is stored as a polymer called glycogen, located in our liver and muscle cells, but that is only enough to keep us going for a few hours, as any runner or cyclist knows. Plants virtually never store fats. they rely on starch instead. Why do plants and animals differ on such a simple feature? Let us look at the consequences of each alternative storage molecule. An energy storage molecule should not be too heavy, and it must be stable enough that it does not go bad within the plant or animal's body. Fat is the most lightweight means of storing energy. A pound of fat, whether it is lard, oil, butter, or margarine, stores more energy than a pound of starch or protein. For an organism that needs to move, weight will be less of a problem if it stores fat rather than if it stores the same amount of energy as starch. The next time your bathroom scale reads 10 pounds more than you want, be glad we do not store starch. We would be even more overweight and have even bigger rear ends. Plants, however, do not move around too much, thus, saving on weight is not a real necessity. For plants, the long-term stability of starch is better than the lightness of fat. Some plants save up energy for years, not just months, and starch will last that long in the plant's body. For example, century plants bloom only once after growing for about 12 to 15 years, and fishtail palms may not bloom until they are more than 70 years old. These plants store some starch each year, then use it all at once in a massive flowering. Fat would not last that long because it becomes rancid if exposed to oxygen, and all parts of a plant are well aerated. Pollen and seeds are exceptions. Many flowers produce pollen with a drop of oil rather than a grain of starch, which makes it lighter and easier for wind or insects to carry it to another flower. Seeds such as peanuts, cashews, and sesame store oil and thus are lighter and smaller and more easily moved by animals. Avocados are very rich in oil, but rather than being an energy storage mechanism, it is a reward that entices an animal to eat the fruit and then spit the seed out somewhere, thereby dispersing the plant seeds far and wide. Plants also store a little starch here and there throughout their bodies, some in cortex cells, some in pith cells, and even wood can store a bit. However, when a plant needs to store a lot of starch, it almost always relies on its roots. The enlarged roots of beets, carrots, radishes, sweet potatoes, and similar plants are filled with starch. By using roots as a storage organ, the plants are putting their reserves underground, out of sight of hungry animals. Also, the soil is a more stable environment, being neither as hot during the day nor as cold at night as the air, and similarly, it maintains a more uniform humidity. The storage tissue in roots is usually wood, a type of xylem. Thus, just as in adipose tissue of animals, root storage tissues are well vascularized. Even though plants and animals store energy differently, the reasons are understandable when we consider the consequences of each alternative. Plants favor stability. Animals need mobility. Mature portions of the root. Root hairs function only for several days, after which they die and degenerate. Absorption of water and minerals in this area is then greatly reduced but does not stop entirely. Within the endodermis, cells may remain unchanged, but usually there is a continued maturation in which a layer of suberin is applied over all radial surfaces, the inner tangential face, and sometimes even the outer tangential face. This can be followed by a layer of lignin and then more suberin. This is an irregular process, and some cells complete it earlier than others. Thus, in fairly mature parts of the root, it is possible to find occasional cells that have only Casparian strips. These cells are called passage cells because they were once thought to represent passageways for the absorption of minerals. It is now suspected that they are merely slow to develop. The result of continued endodermis maturation is the formation of a watertight sheath around the vascular tissues. In older parts of the root, it functions to keep water in. The absorption of minerals in the root hair zone causes a powerful absorption of water, and a water pressure, called root pressure, builds up. If it were not for the mature endodermis, root pressure would force the water to leak out into the cortex of older parts of the root instead of moving up into the shoot. This is presumably also a function of the endodermis in rhizomes and stolons. If water leaked into the stem cortex and filled intercellular spaces, oxygen diffusion would be prevented and the tissues would suffocate. Many important events occur at the endodermis. The cortex and epidermis, aside from root hairs, may be superfluous because shortly after the root hairs die, underlying cortex and epidermis often die also and are shed from the root. The endodermis becomes the root surface until a bark can form. This happens mostly in perennial roots that persist for several years. In apple trees, the cortex is shed as early as 1 week after the root hairs die, although it may persist for as long as 4 weeks. Only the root tips have a cortex. The large fibrous roots of many monocots are strictly annual. They are replaced by new adventitious roots that form on new rhizomes or stolons. In these plants, the entire root dies. Origin and development of lateral roots. Lateral roots are initiated by cell divisions in the pericycle. Some cells become more densely cytoplasmic with smaller vacuoles and resume mitotic activity. The activity is localized to just a few cells, creating a small root primordium that organizes itself into a root apical meristem and pushes outward. As the root primordium swells into the cortex, the endodermis may be torn or crushed or may undergo cell division and form a thin covering over the primordium. As it pushes outward, the new lateral root destroys cells of the cortex and epidermis that lie in its path, ultimately breaking the endodermis. By the time the lateral root emerges, it has formed a root cap, and its first protoxylem and protophlome elements have begun to differentiate, establishing a connection to the vascular tissues of the parent root. Figure 7:16. This young lateral root of willow was initiated in the pericycle, next to a mass of protoxylem. Its growth caused considerable damage to the parent root cortex, and it broke the surface open. Entry of pathogens is prevented by formation of wound bark around the tear it caused. Note how this differs from bud formation in shoots. Lateral roots are initiated deep within the root, not at the surface as are axillary buds. They are formed in mature regions of the root, not right at the base of the apical meristem, and of course, they never develop into flowers. Other types of roots and root modifications. The type of root just described is the most common, generalized type, comprising some or all of the roots of most plants. In other species, some roots are modified and carry out different roles in the plant's survival. Various structures and organizations are selectively advantageous for various functions. Storage roots. Roots frequently provide long-term storage for carbohydrates that accumulate during summer photosynthesis. In biennial species and many perennials, roots are the only permanent organs. In autumn, most of the stem dies back to a few nodes located below ground at the top of the root. Carbohydrates stored in the root are used to produce a new shoot in the spring, when photosynthesis is impossible owing to the lack of leaves. It seems less economical in these species to winter-proof the shoot than to replace it using nutrients stored in the roots. Annual plants can survive without such storage capacity. Perennial plants with permanent shoots that do not die back also store significant amounts of nutrients within themselves during winter, but roots offer certain advantages. Being subterranean, roots are less available as food than are swollen, highly nutritious, easily visible stems. Roots also have a much more stable environment, subjected to less extreme changes in temperature and humidity, which may be important for survival of storage parenchyma cells. Prop roots. The stem of a monocot can become wider, with more vascular bundles, if it can produce adventitious roots that extend to the soil. In some cases, roots are capable of extensive growth through the air. In many palms, the exposed roots can be 20 to 50 centimeters long. In the screw pine, they are often 3 or 4 meters long, and the roots may grow through the air for months before reaching the ground. Less dramatic examples are found at the bases of corn and many other grasses. After prop roots do make contact with the soil, they transport additional nutrients and water to the stem. Just as importantly, they contract slightly and place some tension on the stem, thus acting as stabilizers, much like guy wires on tall television antennas. If the roots undergo secondary growth and become woody, they can be extremely strong supports, permitting a branch to extend even farther from the trunk without breaking or sagging. In banyan trees, prop roots and branches can spread and produce massive trees many meters in diameter. Roots of certain tropical trees become tall, plate-like buttress roots. Their upper side grows more rapidly than other parts of the root. Buttress roots brace the trunk against being blown over by wind, Figure 7. 18b. Figure 7:17. Many monocots like this small palm produce adventitious roots near the base of the stem. These provide both extra absorptive capacity and extra stability. Screw pine produces extremely long adventitious prop roots that not only stabilize the large, heavy trunk but also bring water and minerals into the stem. In older plants of screw pine, even the branches produce adventitious roots. This water and mineral supply to the branches bypasses the trunk, where vascular bundles are already conducting at full capacity. Figure 7:18. Banyan trees produce adventitious roots, which, as in monocots, provide increased support and absorptive capacity. Because the giant branches are supported along their entire length, they can become much larger and more extensive than branches that are supported only at the point of attachment to the trunk. Plate-like buttress roots of Ficus subcordata. These roots emerge from the basal portions of mangrove stems in the Everglades National Park in Florida. Mangroves live in the intertidal zone, the band of seashore between high and low tide. At high tide, the roots are flooded with ocean water. This is a harsh environment because the seawater is so salty it can pull water out of most terrestrial plants and animals, and the water should make the roots drown for lack of oxygen. Mangrove roots survive these conditions because their surface tissues keep seawater away from the delicate cells inside the root, and they have parenchyma that lets oxygen diffuse into the root from the shoots. Mangroves also have prop roots, but these seem to be selectively advantageous for other reasons. The plants grow in intertidal marshes and are subjected to powerful water currents during storms and even normal tide changes. Brace-like prop roots provide much more stability than a taproot system would. In addition, the aerial portion of the prop root is covered with numerous air chambers, lenticels, and its cortex is a wide parenchyma. The subterranean portion of the root grows in a stagnant muck that has little or no oxygen. It is able to respire only because the parenchyma permits rapid diffusion of oxygen from the aerial lenticels to the submerged root tissues. If mangrove roots were entirely subterranean, respiration would be extremely reduced. Few plants have adapted to the intertidal zone. Although ocean shores have existed for billions of years, mangroves became adapted to the intertidal zone only in the last several million years. There are no fossils of mangrove plants older than about 35 million years. Aerial roots of orchids. Many orchids are epiphytic, living attached to the branches of trees. Their roots spread along the surface of the bark and often dangle freely in the air. Although these plants live in rainforests, the orchids are actually adapted to drought conditions. In the few hours when rain does not fall, the air and bark become dry and could easily pull water out of the orchid's roots if there were no water-conserving mechanism. The root epidermis, called a velamen in these orchids, is composed of several layers of large dead cells that are white in appearance. Apparently, the velamen acts as a waterproof barrier, not permitting water to leave the sides of the root. Figure 7:19. Aerial roots of orchids, which grow along the surface of tree bark, are covered by a white velamen that helps retain the water absorbed by the green root tip. These roots grow along the bark, not downward. Contractile roots. In Oxalis, Gladiolus, Crinum, and other plants, roots undergo even more contraction than prop roots do. After extending through the soil and becoming firmly anchored, the uppermost portions slowly contract. Because the root is firmly fixed to the soil, the stem is pulled downward so that the base of the shoot is either kept at soil level or, in the case of bulbs, actually buried deeper. The contraction is caused by changes in the shape of cortex cells. They simultaneously shorten and expand radially, losing as much as 1/2 to 2/3 of their height. The vascular tissues buckle and become undulate but are able to continue conducting. Figure 7:20. The roots of this hyacinth have contracted, causing their surfaces to become wrinkled. Contractile roots may be more common than is generally appreciated. Many seeds germinate at or near the soil surface. Root contraction may be the means by which the shoot becomes anchored in the soil. In bulbs, corms, rhizomes, and other subterranean stems, contractile roots can be important in keeping the stems at the proper depth. Mycorrhizae. Roots of most species of seed plants have a symbiotic relationship with soil fungi in which both organisms benefit. The associations are known as mycorrhizae. And two main types of relationships are known. In nearly all woody forest plants, an ectomycorrhizal relationship exists in which fungal hyphae penetrate between the outermost root cortex cells but never invade the cells themselves. Herbaceous plants have an endomycorrhizal association, in which hyphae penetrate the root cortex as far as the endodermis. They pass through the walls of the cortex cells but cannot pass the Casparian strip. They invade the cell but do not break the host plasma membrane or vacuole membrane. Inside the cell, they branch repeatedly, forming a small structure called an arbuscule. This fills with granules of phosphorus that later disappear as the phosphorus is absorbed by the plant. Other hyphae fill with membranous vesicles. The plant cells lack starch grains, presumably because sugars are being transferred to the fungus. The fungus is unable to live without the sugars from the plant, and in many cases, the plant is severely stunted if the fungus is killed. Apparently, this is a critical means of absorbing phosphorus into the roots, the mycorrhizal fungi being much more effective than root hairs. All the modified roots mentioned to this point, storage roots, prop roots, contractile roots, aerial roots of orchids, and mycorrhizae-associated roots, have a structure virtually identical to that of the more common anchoring absorbing roots described above. All grow by an apical meristem, and a cross-section through a mature region of each reveals epidermis, cortex, endodermis, pericycle, and vascular tissue. In nitrogen-fixing nodules and haustoria, however, the structure is highly modified. Root nodules and nitrogen fixation. For most plants, the scarcity of nitrogenous compounds in the soil is one of the main growth-limiting factors. Although nitrogen is abundant in air, plants have no enzyme systems that can use that nitrogen. Only some prokaryotes can use N by incorporating it into their bodies as amino acids and nucleotides. When they die and decompose, the nitrogenous compounds are available to plants. The chemical conversion of atmospheric nitrogen into usable compounds is nitrogen fixation. Figure 721 diagram of endomycorrhizae showing hyphae penetrating the root cortex and forming arbuscules and vesicles. Root cells filled with fungal hyphae in the form of arbuscules. You can tell that this is a symbiotic fungus rather than a pathogenic one because the plant cells are healthy, with large normal nuclei. Roots of beech encased in ectomycorrhizae. The presence of the fungus stimulates the roots to be short and broad. Transverse section of an ectomycorrhizal root showing that the fungal filaments are restricted to just the outermost part of the root, they do not penetrate the cortex. In a small number of plants, especially legumes, a symbiotic relationship has evolved with nitrogen-fixing bacteria of the genus Rhizobium. Bacteria free in the soil secrete a substance that causes root hairs to curl sharply. A bacterium then attaches to the convex side of the hair and pushes into the cell by means of a tube-like invagination of the plant cell wall. The tube is an infection thread, and the bacterium sits in it. The infection thread, penetrating one cell after another, extends all the way into the root's inner cortex, where adjacent cortical cells undergo mitosis and form a root nodule. The bacterium is released from the infection thread and enters the plant cell cytoplasm, where it proliferates rapidly, filling the plant cells with bacterial cells capable of converting N into nitrogenous compounds that are released to the plant cells. Energy for this process is supplied by sugars from the legume root cells, so both the rhizobium and the legume benefit. The nodule may remain rather simple or may become complex, with a meristematic region, vascular tissue, and endodermis. Such nodules function for extended lengths of time. Numerous metabolic changes have also evolved. The critical bacterial enzymes are sensitive to oxygen, being immediately poisoned by even traces of free oxygen. However, it is the plant, not the bacterium, that produces a special chemical, leg hemoglobin, that binds to oxygen and protects the bacterial enzymes. Root nodules represent a sophisticated symbiosis. Plants and bacteria both benefit. The bacteria are not damaging the plants, nor are the plants parasitizing the bacteria. Without the bacteria, the complex development of nodules does not occur. This process is often described in this way: the bacterium supplies the plant with nitrogenous compounds and in return the plant gives it sugars. This inaccurately suggests voluntary action, choice, and decision-making on the part of both organisms. Instead, think in terms of natural selection. Because the bacterium receives sugars and other nutrients from the plant, any genetic mutation in the bacterium that makes the plant healthier and more vigorous is beneficial to the bacterium as well. Whereas any mutation that is harmful to the plant, such as a bacterial plasma membrane that does not allow nitrogenous compounds to move to the plant, is deleterious to the bacterium. The same principle applies to the plant. Genetic mutations that aid the bacterium ultimately benefit the plant as well. We hypothesize that these organisms will continue to co-evolve and become more fully co-adapted, but this hypothesis will have to be checked by someone a few million years from now. Figure 7.22. The nodules on these alfalfa roots contain bacteria capable of absorbing atmospheric nitrogen and converting it to ammonia, which the plant can use to make amino acids. Bacteria in an infected root nodule cell of cowpea, Vigna unguiculata. Working backward, we can imagine less sophisticated legumes and rhizobium bacteria that did not work so well together but evolved to the present state as mutations occurred and survived by natural selection. Certain critical mutations must be rare, or perhaps several unusual mutations must occur almost simultaneously in plant and bacterium. This hypothesis is based on the observation that, although this symbiosis would probably be beneficial to virtually all plant species, only a few have it. Hostorial roots of parasitic flowering plants. A number of angiosperms are parasites on other plants. Because their substrate is the body of another plant, a normal root system would not penetrate the host or absorb materials from it effectively. Consequently, roots of parasitic plants have become highly modified and are known as hostoria. In most cases, very little root-like structure remains. Parasitism has evolved several times, thus, the structures that are termed hostoria are not all related to each other, and generalizations are difficult. Hostoria, however, typically must adhere firmly to their host either by secreting an adhesive or by growing around a small branch or root. Penetration occurs either by forcing a shaft of cells through the host's dermal system or by expanding the hostorium radially, cracking the host's epidermis. After penetration, cells of the parasite make contact with the host's xylem. In many cases, both host and parasite cells divide and proliferate into an irregular mass of parenchyma, and then a column of cells differentiates into a series of vessel elements. This results in a continuous vessel from host to parasite constructed of cells of both. Surprisingly, many parasites attack only the xylem. They do not draw sugars from the host but carry out their own photosynthesis. Others do contact both xylem and phloem and perform little or no photosynthesis. For such parasites, mutations that result in the loss of leaves or vegetative stems are not disadvantageous. Figure 7.23. Transverse section of a branch of juniper being attacked by the hostorium of a mistletoe. The wood of the host juniper has been stained blue, the bark is tan. The hostorium is able to draw water and nutrients from the host vascular tissue. A parasitic plant named Tristrix spends most of its life as nothing more than a diffuse root system, a hostorium, growing inside the body of its host plant, a cactus. The body is just a network of parenchyma cells with no stem, leaves, epidermis, cortex, or vascular tissues except when Tristrix produces flowers. The typical organization of a root would be non-functional in hostoria. The root cap, root apical meristem, and cortex would all prevent vascular tissues of the parasite from contacting the appropriate tissues of the host. Conversely, hostoria are completely inadequate for growth in soil, however rich and moist it might be. For each substrate and each microhabitat, specific adaptations are selectively advantageous. Roots of strangler figs. While young, plants of strangler figs grow as epiphytes perched on a branch of a host tree. Birds eat fruits of strangler figs and then deposit the seeds on branches of other trees. When the strangler fig seed germinates, its roots cling to the bark of the host tree branch and then grow rapidly downward, hugging the trunk of the host tree. For some months or even years the roots have no contact with the soil, so they must absorb nutrients from rainwater that runs down the trunk of the host tree. Finally, the strangler fig's roots do reach soil and then penetrate it rapidly and branch profusely. Upper portions of the roots enlarge and become woody. Strangler fig roots branch and grow at various angles, encircling the host tree's trunk, and the roots fuse to each other wherever they meet. Before long, the trunk of the host tree is encased in numerous roots of the strangler fig. Typically, the host tree finally dies and rots away, leaving the strangler fig as a self-supporting tree with its root-shoot junction high in the air. It was believed that the tight network of strangler fig roots prevented the host tree from growing thicker, and thus the strangler fig killed the host by strangling it. That may be true in some cases, but it seems that more often the fig produces so many leafy branches that it shades the host tree so much it cannot photosynthesize. The host tree dies from lack of light. Figure 724. This tree in the Everglades National Park is encased in the roots of a strangler fig. The large branch of the fig at the upper left is actually the trunk of the fig, which is the point where the seed germinated. All portions below that are roots of the strangler fig. It would be easy to mistakenly assume the fig is a vine and the parts encasing the host tree are stems of the fig, but a close examination of the anatomy shows these are roots, not branches. If this were really a vine, we would expect to see leafy branches emerging from many areas in this photo. At the next level. 1. Modified roots. This chapter touched briefly on root nodules and nitrogen-fixing bacteria and also on mycorrhizal associations of roots and phosphate-absorbing fungi. Much more is known about both these topics, and you can learn a great deal by exploring on your own. Other modified roots for you to investigate are the coralloid roots of cycads and proteoid roots. Roots of many epiphytes are modified such that their growth is not controlled by gravity but instead is guided by contact with host tree bark. 2. Root ecology. Root systems vary greatly in size, branching, orientation, and so on. Some roots spread horizontally in the topsoil, others grow downward 30 m or more. Certain plants have much more root mass than shoot mass, whereas some large globular cacti have only a few scrawny, thread-like roots. We are starting to understand the factors that influence root growth, so this is a rich area for you to explore. Especially important are the enormous root systems of prairie grasses. 3. Parenchyma and flooding. Many river valleys flood during springtime, causing roots to be flooded for days or weeks. This reduces the amount of oxygen available, and the risk of suffocation is great. Some roots and stems respond by producing parenchyma under such conditions, allowing more rapid diffusion of any oxygen present. Marsh plants such as mangroves and bald cypress automatically produce knees, technically called nematophores. Pronounced nematophores, modified parenchymatous roots that grow upward into the air. Summary. 1. Most roots have a variety of functions, including anchorage, absorption, and hormone production. Other roots may be specialized for nutrient storage, vegetative reproduction, or surviving harsh conditions, some even being modified into spines. 2. Roots have a root cap but nothing equivalent to leaves, nodes, internodes, or buds. 3. Adventitious roots form in organs other than other roots or the embryo. They are especially important in stoloniferous and rhizomatous plants. 4. Roots, like shoots, elongate by localized growth. Only the root tip and zone of elongation must slide between soil particles. 5. Root hairs greatly increase the absorptive surface area of the root system, and the carbonic acid that results from their respiration helps release minerals from soil particles. 6. An endodermis with Casparian strips prevents minerals from moving from the soil solution into the xylem. To enter the xylem, minerals must at some point cross a plasma membrane. 7. Lateral roots arise in the pericycle, deep inside the root, unlike axillary buds and stems, which arise in the outermost stem tissues. 8. Prop roots provide additional stabilization and transport for certain plants with narrow stems. Contractile roots aid in burying certain bulbs, corms, and rhizomes. Haustoria are modified roots that penetrate the tissues of host plants. 9. Most plants absorb much of their phosphorus from mycorrhizal fungi that form an extensive network both in the soil and within the root cortex. 10. A small number of plant species form symbiotic relationships with nitrogen-fixing prokaryotes. The bacteria or cyanobacteria often live in root nodules, passing nitrogen compounds to the plant and receiving carbohydrates and minerals. Important terms: adventitious roots, branch roots, buttress roots, Casparian strips. Ectomycorrhizal relationship, endodermis. Endomycorrhizal association, fibrous root system, Hostoria infection, thread lateral roots. Mycorrhiza nitrogen fixation, pericycle, prop roots, quiescent center radical. Root apical meristem, root cap, root hairs, root nodule, taproots, zone of elongation. Review questions. 1. The concepts section of this chapter states that most roots have 3 functions. What are they? Some roots have other functions in addition to these 3, or instead of them. Name an example of at least one species in which roots do the following. A. Store carbohydrate during winter. B. Produce shoot buds that can act as new plants. C. Grow out of the trunk and harden into spines. D. Act as holdfasts. E. Attack other plants and draw water and nutrients out of them. 2. What are the 2 types of root systems? Give several examples of plants that have each type. Which type is associated with nutrient storage in biennial species like carrots and beets? Which is associated with rhizomes and stolons? 3. Roots must have an enormous absorptive surface. Why do plants have a highly branched root system instead of just one long root? 4. Even before a seed germinates, it already has a root. What is the name of this embryonic root? In eudicots, what does this embryonic root usually develop into? In most monocots, this embryonic root does a strange thing during or immediately after germination. What does it do? 5. What is an adventitious root? In a monocot rhizome such as that of irises and bamboo, are adventitious roots common or rare? If a bamboo rhizome grows 100 feet underground and then sends up an aerial branch, where does that branch get its water? From the roots 100 feet away or from adventitious roots right at its base? 6. What does the root cap do? Do you think this structure would have evolved if roots all grew in air like shoots do, rather than growing through dirt? 7. Roots have localized growth. The only parts of the root that become longer are the root apical meristem and the of, a region only a few millimeters long within which the cells undergo division and expansion. 8. What would happen to root hairs if they formed in the zone of elongation? Would this happen if roots were growing in air or in water? 9. Root hairs greatly increase the root's surface area. In rye, a single plant has been found to have lateral roots with km of root length and a surface area of m. Convert the length to miles and the area to square feet. 10. Root hairs are narrower than roots. A root is usually at least mm in diameter, but root hairs are only approximately mm in diameter. Look at figure 7-9. Could either roots or root hairs enter the pores represented by the red part of the chart? Could either or both enter the gray part? The white part? Most roots grow near the soil surface, in the upper 20 centimeters, where most of the soil pores are between 0.2 to 60 millimeters wide. If a root did not produce any root hairs at all, could the plant obtain water and minerals from these abundant, narrow pores? 11. Which part of the root detects gravity? Which organelles enable them to do this? 12. The use of a radioactive precursor of DNA can demonstrate that the central cells of the root apical meristem are not synthesizing DNA. This mitotically inactive central region is called the. It is now believed that the central cells act primarily as a reserve of. 13. If you could examine a transverse section through the zone of elongation in a root, would you see any mature cells? Would there be any fully differentiated epidermis cells or vascular tissues? What would the outermost cells be called, if not epidermis? In the center of the zone of elongation, what would be the tissue that would later differentiate into xylem and phloem? 14. Examine figure 7.13. What are the 4 diffusion paths that a molecule might follow as it travels through the root epidermis and cortex? Why does this diagram stop at the endodermis? What happens at the endodermis? 15. describe the shape of the endodermis. Is it a flat plane, a cylinder, a single ring of cells? What are the Casparian strips, and on which cell walls are they located? 16. Draw cross-sections of a root showing its structure at 3 levels, the mature region, the root hair zone, and the zone of elongation. At which level is the endodermis complete with Casparian strips? 17. Because Casparian strips are impermeable, minerals can cross the endodermis only if the endodermal absorb them from the intercellular spaces or from cortical cells. Many minerals can be excluded by the endodermis. 18. Examine figure 7.14. The roots of eudicots are usually noticeably different from those of monocots in transverse section. Which has a relatively wide set of vascular tissues? Which has endodermis cells with very thick walls? Which has ground tissues in the center, and which has metaxylem in the center? 19. Which part of the root produces the primordia for lateral roots? How does the vascular tissue of the lateral root connect with that of the parent root? 20. Some perennial plants store significant amounts of nutrients in the stem during winter, but roots offer certain advantages. Describe 2 of these advantages. 21. Animals typically use fats in adipose tissues for long-term energy storage, whereas plants use starch in roots. How do animals benefit from using fat? How do plants benefit from using starch? Name 2 plants that store energy for many years. How long is long-term storage for these species? What 2 plant parts often use fats and why? Storage tissue in enlarged roots is vascularized. How is that important to the plant? 22. Look at the prop roots of screw pine in figure 7.17d. Because the branches produce their own adventitious roots, can the branches obtain water and minerals without depending on the xylem in the trunk? 23. Look at the prop roots of banyan trees in figure 7.18. These prop roots supply extra water to the branch, but how else do they help the branches? 24. What are contractile roots? They are especially common in plants with what types of shoots? 25. What is a mycorrhizal association? What benefit does the plant derive from the association? 26. Describe the structure of a nitrogen-fixing nodule. Consider especially the relationship with the plant's vascular tissue. 27. Root nodules are associations. Plants and bacteria both benefit. The bacteria are not the plants, nor are the plants the bacteria. 28. Many plants are parasitic on other plants. These parasitic plants have modified roots called. Describe some of the ways that this parasitic root penetrates the host stem or root. Do these parasitic roots have a root cap, a root apical meristem, or a cortex? 29. Except when it produces flowers, what is the body of the parasitic plant Tristerix? What tissues does this plant body lack, except when it produces flowers? 30. Imagine a plant that has 10 roots, each 1 centimeter long. What is the total length of the root system? Imagine that at the tip of each root are 10 lateral roots, each 1 centimeter long. Now what is the total length of the root system? What is the maximum distance a water molecule must travel from the farthest root tip to the base of the shoot? Distance traveled increases as a simple addition, but total absorbing capacity increases exponentially. Why is the length that is traveled important? Consider the friction of moving through tracheids and vessel elements. Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated ZArt. Chapter 8. Structure of woody plants. Chapter opener image. These are relatively small trunks of western red cedars. They become more massive with age. Each year, the trunk, branches, and roots add a new layer of wood and bark, making them stronger and more conductive to water, minerals, and sugars. With such strength, the trees grow tall enough not only to put their leaves above those of shorter plants, they also put their reproductive cones higher, such that there is a better chance wind will carry pollen from each tree to many others. Outline. Concepts. Vascular cambium. Initiation of the vascular cambium. Fusiform initials. Ray initials. Arrangement of cambial cells. Secondary xylem. Type softwood cells. Growth rings. Heartwood and sapwood. Reaction wood. Secondary phloem. Outer bark. Cork and the cork cambium. Lenticel sand oxygen diffusion. Initiation of cork cambia. Secondary growth in roots. Anomalous forms of growth. Anomalous secondary growth. Unusual primary growth. Box 8-1 Botany and Beyond, wood in 3 dimensions. Box 8-2 Plants do things differently, having multiple bodies in one lifetime. Box 8-3 Plants and People, dendrochronology, tree ring analysis. Box 8-4 Plants do things differently, thinking about the growth of wood. Learning objectives. After reading this chapter, students will be able to explain the advantages and disadvantages of secondary growth. Define the vascular cambium, fusiform initials, and ray initials. Describe the arrangement of cambial cells. Discuss the axial system of wood. Identify the portions of growth rings. Define heartwood, sapwood, and reaction wood. Compare secondary phloem to secondary xylem. Contrast cork cambium and vascular cambium. Explain the function of lenticels. Discuss secondary root growth. Name 3 examples of anomalous secondary growth. Give an example of establishment growth. Did you know? Woody plants are usually easy to recognize. If you think a tree or shrub is woody, you are probably correct. Only a few plants will fool you. Palms and bamboos have no wood, and many plants that appear to be herbs have a small amount of delicate wood. Most woody plants add a new layer of wood around their trunk, branches, and roots every year. There is no theoretical limit to how long a plant can live and how wide it can become. A cypress in Oaxaca, Mexico has a diameter of 11.6 m. Concepts. Growth by means of apical meristems has been previously described. From the meristems are derived sets of tissues, epidermis, cortex, vascular bundles, pith, and leaves. These primary tissues together constitute the primary plant body. In plants known technically as herbs, this is the only body that ever develops, but in woody species, additional tissues are produced in the stem and root from other meristems, the vascular cambium and the cork cambium. The new tissues themselves are wood and bark. They are secondary tissues, and they constitute the plant's secondary body. Examples of woody plants are abundant. Trees such as sycamores, chestnuts, pines, and firs are woody, as are shrubs such as roses, oleanders, and azaleas. The ability to undergo secondary growth and produce a woody body has many important consequences. In an herb, after a portion of stem or root is mature, its conducting capacity is set. All provascular cells have differentiated into either primary xylem or primary phloem. This capacity is correlated with the needs of leaves and roots. If the plant produces so many leaves that they lose water faster than the stem xylem can conduct, some or all the leaves die of water loss. Similarly, it would not be selectively advantageous for the plant to have so many leaves that they could produce sugar faster than the phloem could conduct it to roots, flower buds, or developing fruits. Many herbs live for several years, however. How do they respond to the bottleneck of the limited conducting capacity of the first-year stem? In some, the first year's leaves die during winter, and in the second year, the plant produces only as many leaves as it had during the first year. In other species, adventitious roots are produced that supply conducting capacity directly to the new section of stem being formed, thus bypassing older portions of the stem. Most of these plants must remain low enough for adventitious roots to reach the soil. These are often rhizomatous, such as irises, bamboo, and ferns. Woody plants not only become taller through growth by their apical meristems but also become wider by accumulation of wood and bark. Because wood and bark contain conducting tissues, their accumulation gives plants a greater capacity to move water and minerals upward and carbohydrates downward. The number of leaves and roots that the plant can support increases, as does the photosynthetic capacity. For example, consider a tree with a trunk radius of 10.5 centimeters. Imagine that the plant produced a layer of wood 0.5 centimeters thick in the